Okay, this lesson is going to be on the process of protein synthesis. So, how we go from DNA to protein. So, we want to make sure we can identify the different types of RNA because there's three different types that we're going to talk about, and they're all play a very important role in protein synthesis. And then we're also going to just describe the steps of transcription and translation. So, three types of RNA. We have our messenger RNA or mRNA, ribosomal RNA, and transfer RNA. So, our messenger RNA, this is the actual RNA that carries the information about a specific protein. This is what's transcribed from DNA, and um, three of your mRNA bases form a codon, and it's these codons that are going to actually specify the specific type of amino acid. And so those amino acids are what's going to be the building blocks of our proteins. Our ribosomal RNA, this is important because it basically is what forms the ribosome. So this is what plays a super important part in translation that we're going to talk about in a little bit. And it basically functions as a machine that's going to link those amino acids together. Our transfer RNA or tRNA, it gets its name not only because of its function, but because of the um, kind of the shape of the protein of the RNA. Um, But this is what actually carries those amino acids around. There's specific parts of this molecule that we're going to label later on, but it's important um, to know that there's an anticodon section. This is the part that's going to be complementary to the mRNA, and then there's also another end where that amino acid is going to actually bind. And this is just a picture. So we have our mRNA. This looks very similar to our DNA. It's just basically the sequence of base pairs. Our ribosomal RNA. This is what forms our ribosome. So we have a large part of our ribosome, and then we also have the smaller portion. And then here's our tRNA. So this is what I meant by it kind of looks like a T. The way that it kind of folds in on itself because it has these complementary base pair structures um, that it actually can kind of folds in and they bond together and it forms this kind of T structure. So down here in the bottom, you have this what we call an anticodon. This is what's actually going to recognize our mRNA sequence. And then in the top, we have our section where the amino acid is going to bond. So there's a central dogma that kind of goes along with molecular biology and genetics. It basically describes this directional flow of genetic information. So it states that there's genes in our DNA that specify a sequence of mRNA, which then specifies a sequence of proteins. So it's a kind of unidirectional process of um, I guess, gene translation. Um, so first, we're going to talk about transcription. There's three kind of separate parts. We have our initiation, elongation, and termination. The first part has to do with this kind of initiating this um, of the actual transcription. So we have our DNA sequence here, um, and we have this kind of starting sequence, I guess you could say, or it's called a promoter sequence. So you usually have a large number of these kind of repetitive G's and C's, and then right in the middle you have what we call sometimes it's called a TATA box or a TATA box. Um, this is kind of the signal that this is where a gene is about to start, or this is where a gene sequence is beginning. So we have a transcription factor that's actually going to bond to this section's promoter sequence right here. And that transcription factor is what kind of signals the RNA polymerase to begin transcribing the DNA. And it transcribes in the same direction that DNA polymerase does. It actually moves along the DNA strand from the 3 to 5 prime direction, and then it forms RNA from the 5 to 3. So remember, because it's still kind of has that anti-parallel facet with it. So in our elongation, this is where we're just continuing to read those nucleotides. So there's enzymes that are unwinding that DNA double helix. Those free nucleotides are bonding. So the RNA polymerase is instead of adding, it kind of functions very similar to DNA polymerase, except for we're swapping out our T and our U. And then the terminator sequence, there's actually just like we have kind of that promoter sequence at the beginning of the gene, there's a terminating sequence at the end of the gene, and that kind of signals for the RNA polymerase to fall off. So here's just a little image of what we just talked about. So we have our little promoter region where our RNA polymerase eventually will bind. As it's moving along, it's transcribing mRNA. Eventually, you can see it's made its way down, and eventually it's going to just kind of fall off once it reaches that terminating sequence. So once mRNA has been transcribed, um, remember, all this is taking place in the nucleus where DNA is located. However, our protein synthesis actually takes place in the cytoplasm. So somehow our mRNA has to actually exit the nucleus and enter the cytoplasm. In order to do that, there's a couple of steps that need to be done to kind of protect this mRNA while it's in the cytoplasm. So the first thing that's done is there's a methylated cap. So it's basically just some um, carbon structure is added to the 5' end of the mRNA. And this is what's going to kind of help the mRNA bind to or attach to the ribosomes for translation. And then a poly A tail is added to the 3' end. Um, and this tail kind of protects this strand from any kind of digestive enzymes in the cytoplasm. The second thing that occurs is something called splicing. So we've discussed the terms introns and exons before, and these introns are kind of this just these sequences inside a gene that aren't really important. They don't really um, code for anything. They're just in there. So those sections are actually going to be removed from our mRNA. And then the exons, they're spliced together via something we call spliceosomes. And this is what forms our mature RNA or our secondary mRNA transcript. So this is kind of that final mRNA transcript that can then move on to be translated. So here's just kind of a picture. So we have our DNA, right? And we have our a couple of introns in here that we know are not important. They're still transcribed because there's no way to really, for the RNA polymerase to really know where those are. Um, and so they're not going to be taken out until you get kind of down here in this splicing section. So you can see we've added this cap. It has its poly A tail. Poly A tail is just kind of a string of A bases. And then now our splicing, we can see that we've kind of removed these two introns. So here we have our mature mRNA, which is going to actually code for some sort of protein. So translation, our mRNA has exited the nucleus and it's kind of bound to this ribosome. And what happens is this mRNA strand is going to kind of, as it moves through the ribosome, that's what's going to, these tRNAs are going to kind of come in and add these amino acids together. And there's a specific way that this occurs. Before we get into the steps of translation, though, I think it's important to kind of go over a couple of facets about the structure of some of these RNA molecules. So if we're looking at tRNA, each one of these tRNA segments or molecules is specific for a particular amino acid. Up here at the top, we have what we call the anti-codon. So this is just the complementary sequence to our mRNA. So this is what's going to bind to that codon on the mRNA. On the other end, this is where we have our amino acid. Okay, so you can see here we have a leucine that's bonded to this GAU anticodon. For our ribosomes, our rRNA, there's some important kind of sections to our ribosome that's important. So we have different sites. Our A site is kind of where our tRNA kind of first enters the ribosome. So this is where it's first going to attach to our codons on our mRNA strand. Okay, this tRNA, remember, is carrying an amino acid. As the mRNA kind of moves through the ribosome, it's going to slide down to where that tRNA is now in our P site. Okay, the P site is where this amino acid is going to attach to our growing chain, and that's kind of where the chain continues to form. Then, once the mRNA moves down one more time, that's where our tRNA is going to kind of exit our ribosome. So for our initiation steps, we have a start codon, which is this AUG. This is the actual mRNA sequence, AUG. So our anticodon is UAC. This is the first tRNA that's going to actually bond here. This is what signals that it's kind of the beginning here, and it kind of helps make sure that it's properly positioned inside the ribosome. Okay, so our starting codon here is always going to be AUG. And then, like we kind of said, as the mRNA kind of moves through the ribosome, just kind of segmenting down, all these tRNAs are just kind of coming in, binding to that A site, and then just moving down with the mRNA. So they move into the P site where the amino acid chain or polypeptide chain is extended, and then they move on to the E site where that tRNA is going to exit the ribosome. And it is important, I think, to know how these amino acids are bonded together. This picture, you don't necessarily have to know the structure, but you do need to know that it's called a peptide bond. That's why these are called polypeptides. They're held together by peptide bonds. So it just kind of has to do with, because we have a negative charge here and a positive charge over here, they can actually bond together. So they share some electrons here. The end of translation occurs because your mRNA strand actually has a stop codon, and there's 3 different stop codons. They're listed here in this picture. So as soon as the tRNA reads the stop codon, it no longer um, it doesn't actually bring in a new amino acid. It just releases. So that just kind of signals the release of that mRNA sequence from the ribosome. This is kind of the chart of the different amino acids. So I think it's important to note here that there's several different codons, all code for the same amino acid. So there's actually 64 unique codons, and they all code for 20 amino acids. So some of them are going to be repetitive. Um, One thing about the genetic code is it's redundant. That's what I was saying. How there's multiple codons that all code for the same amino acid. So there's four. We have four codons here that code for leucine. There's two more up here that code for leucine. Um, there's a couple of lysines here, glycine. Okay, so it's not just one codon per amino acid. And the other really important part of this is that it's as universal for all living organisms. So um, these same codons code for these same amino acids in every living organism. It's not specific for humans. It's not specific for mammals. It's all living things. This is a good summary video. I'm not going to play it here because the sound is going to do a weird echo, but I will post this link to Schoology so you can kind of go through and watch it after the notes. I think it gives a good visual of all the steps and then kind of a good description or summary of everything as well.